talk about the practical that you need to conduct. In this, we are going to talk about a few types of eggs, their cleavage, and further stages in embryogenesis. To begin with, we need to get acquainted with the egg cell or the oocyte of a mammal. That is human oocyte we will take as typical example. When I say oocyte, it is not the same as an ovum, particularly for human beings. Because for human beings, the egg cell which is given out, which is released out from the ovary, is yet to mature. It's actually a stage called secondary oocyte which is given out. So we better call it as an oocyte, which is still to develop into an ovum, which happens at the time of fertilization. So what you're seeing here is an ovum which is yet to mature. So it's preferably known as an oocyte. Now this oocyte which has been released out from an ovary is the cell, haploid cell with a haploid nucleus. The covering is called as white-lined membrane. That's covering of the egg cell. That is secondary oocyte. Around it, there's one more thick covering. A jelly-like layer, which is called as zona pellucida. Now this zona pellucida has a prime function of protecting the egg, nourishing it, and very importantly, when it is moving through the fallopian tube towards the uterus, this covering sees to it that the eggs doesn't stick to any other position. It straight away moves into the uterus. In addition to this, there are a few diploid follicular cells. These are the follicular cells that I am drawing. These are the follicular cells, and the layer, it's called as corona radiata. These are the diploid follicular cells. So now the oocyte or the egg cell in a human being or a typical mammal would be a haploid egg cell, haploid nucleus as I mentioned, with a covering that's called as white-lined membrane, a jelly-like layer around it that's called as zona pellucida, and a cellular layer covering it is corona radiata. This is the egg cell which is given out from the ovary into the fallopian tube or into the coelomic cavity, from where it is collected into the fallopian tube. This is going to get fertilized. Now, in the process of fertilization, the sperm which has to enter it can pass through the cells, through the follicular cells, enter through the zona pellucida, but it cannot directly penetrate the white-lined membrane. So the covering has to be penetrated with an appropriate method. And the sperm, if you recollect what we discussed in the theory, has a cap which is called as an acrosomal cap that contains an enzyme called hyaluronidase, or sperm lysin it is called. So the sperm comes in, opens its acrosome, so the sperm lysin is given out here. But that makes a small dent, small depression in the covering. That sperm is dead now. Second sperm comes in, it does the same thing, the depression becomes deeper. Third sperm, so number of sperms keep coming and opening their acrosomal caps here. Finally, there is a sperm which opens its acrosome, and that leads to formation of a small hole in this, through which the sperm can enter in and bring about the process of fertilization. That is how it happens. So before a sperm is able to enter in, a number of sperms have actually broken open their acrosomal caps to make the final hole there. This is the oocyte, or an egg cell. What does a sperm look like? Actually, we have seen it in one of the earlier sessions, but still just a revision. It has this is the structure of the tip. This is the haploid nucleus. This headpiece, midpiece, tail, which should be much longer. It continues here, and in this region there would be a number of mitochondria. Here is the proximal centriole, here is the distal centriole, this is the axial filament that then continues as the end piece. So this is the acrosomal cap, you can see. This is the nucleus, haploid nucleus. Here is the proximal centriole. These are the mitochondria. This is the distal centriole. Here is the axial filament. This would be the headpiece, midpiece, tail, and the end piece. That is the structure of the sperm. Functions of different parts we have already discussed. Acrosome is responsible for rupturing the outer covering of the egg cell so that the sperm can undertake fertilization. Nucleus to carry the haploid set of chromosomes from male into the female cell. Centrioles help in mitotic divisions of the zygote. Mitochondria for generating energy that's required for movement of the sperm. Axial filament is a kind of flagellum that helps in movement of the tail actually. So that's the mammalian sperm, that's the mammalian egg cell. Let us have a look at the fish egg cell or fish egg. The first thing that we must understand that it's a megalithical and teloleucithal egg. That is, this could be the egg cell, wherein major part of the cell is occupied by yolk. So this could be all yolk. It has an additional covering. This is called as a chorion. This is the vitelline membrane. As I already said, it's a megalocecal egg, so large amount of this is occupied by yolk, which would mean this would become the animal pole, this would become the vegetal pole. Part of the egg cell occupied by yolk, part of the egg cell containing cytoplasm and nucleus. That's how we can actually identify them. This is what the fish egg would be looking like. Chorion is a protective layer. Vitelline membrane is a cell membrane that actually has some role in the fertilization process. Frog egg, when we discuss. Here also, this could be the egg cell, nucleus, cytoplasm. Now here what happens, this is also megalocecal, this is all yolk, this is all cytoplasm and nucleus. So this will be vegetal pole, this is animal pole, and here there is a small region which is demarcated as gray crescent, which would play a vital role in the process of development of an embryo. So here also animal pole and vegetal pole are clearly distinguishable. In this, there is an additional layer around it, a thick covering, that is a jelly layer. Jelly layer, because see, in frogs the fertilization is external. The eggs are laid out into the water, the sperms are shed over it, and that is where the fertilization has to occur. So this works as a kind of protective layer. That also helps the eggs to stay together. That's called as a spawn, and that spawn stays attached to a suitable substratum. All that is possible because of the jelly layer that's around the egg cell. This is what the frog egg looks like. This is the chick egg, a bird egg, let's say, which, as you all must be knowing, it's macroleucithal or largely macroleucithal. Large amount of the egg is occupied by the yolk. Let's understand the different parts of it. This is the outermost covering, as we commonly call it to be the shell, the calcareous outermost covering, inside which there are two layers: the outer shell membrane and inner shell membrane. Now, major part of this is occupied by the egg white or the albumin. This is outer egg white, outer albumin. This is inner egg white or inner albumin. Then this is the egg cell which is covered by vitelline membrane, inside which, see, as I've already said, it's a largely macrolysthal. So rest all what you are seeing here is nothing but the egg yolk, which is actually two types of yolk. As you are seeing, there are two layers, circular layers. These are yellow yolk and white yolk alternating with each other. Then you are seeing a flask shaped structure that's called as the latebra. Latebra, wherein yolk has accumulated to a considerable extent. This is the nucleus. And rest all would be the cytoplasm. Here you are seeing the egg undergoes a kind of twisting, and these are the strands which are formed out of that twisting. These are called as the chalazae. Chalaza is singular. Okay, this is what the bird egg looks like. Now, mind well, here the cleavage, the division is going to be discoidal cleavage. Only in this region there will be cleavage, and that too, it is not holoblastic, it is meroblastic cleavage, partial cleavage. Rest of the structure is not going to divide. There's a small air sac here that helps the developing embryo. Now, which way does it help? We will get to understand a little later. Now, this is what the chick egg looks like. Mind well, it's a cladoic egg, so it has a hard calcareous covering, the shell. Okay. Let's then talk about the different types of cleavages in specific examples. With specific examples, we will consider different types of cleavages. Suppose to start with, we consider Amphioxus. Mind well, Amphioxus is microlysthal egg, contains a very little amount of yolk, and that too it is evenly distributed. So, if, see, this is the fertilized egg, say the zygote, then the cleavage is, first cleavage is meridional, second cleavage is also meridional but perpendicular to the first one, fourth cell is on the other side, third cleavage is latitudinal. But every time, as you are able to see, these cells form, the daughter cells are formed of the same, they are of the same size and shape, of course. So this is holoblastic, that is complete and equal cleavage. The daughter cells formed, the blastomeres formed are of the same size and shape. So it is holoblastic equal cleavage. If we talk about, say, frog, first thing that we must understand, in the case of frog, half of the egg is cytoplasm and nucleus, rest of the egg is yolk. So it is a telomerase. egg, macrolecital and telolecital. That is large amount of yolk and that too accumulated in one half of the cell, one side of the cell. So when it undergoes cleavage, again the first cleavage is going to be meridional. Yolk would be as it is. Second cleavage is again meridional. Third cleavage now is horizontal. Horizontal, we are not calling it latitudinal because it's not exactly in the half. It would be like so. See, this is animal pole, this is vegetal pole. So the cells formed in the region of animal pole, these are the cells. We will call them as micromeres. Those formed in the region of vegetal pole, they would be megameres. So you can clearly understand the difference in the size. So this time the cleavage is holoblastic, that is complete but unequal. The cells formed will not be of the same size and shape. Micromeres and megameres are clearly distinguishable. That is what you see in the case of frog. Let's understand the cleavage in a bird, maybe a chick. Now here, as we have already learnt, it's a largely megalicidal egg cell. It contains huge amount of yolk. So if say this is the egg cell, major part of this is occupied by the yolk. Cytoplasm and nucleus would be on one side. This is all yolk. So whatever cleavage, whatever division it has to undergo, it would be only related to this part. Hence the cleavage that happens here, it's called as Antiscoidal cleavage, and that too it is meroblastic, which means the furrow that divides the cell, that divides the zygote, doesn't completely pass through the cell. It partially passes through the cell, the rest of the cell remains almost undivided. So it is meroblastic and discoidal cleavage that is found in birds, which then leads to formation of a blastula, which is called as a discoblastula. So that's the kind of cleavage. Suppose if you see it in the top view, this is the cytoplasmic component, this is the rest of the cell with yolk. The cleavage would occur only in this part. That is the kind of cleavage you would find in bird. Whereas in the case of bird, as we have seen the structure, this is the region where all the cytoplasmic contents will be present. Rest all is albumin and all. In this, the part that contains cytoplasm and yolk only would be undergoing division. Rest of the region will not divide. So the cleavage would be like meroblastic. The cell doesn't completely divide. Partial division of the cell would take place. So it's a meroblastic cleavage. Rather, the embryonic development would be related to this disc. Okay, so a disc-like structure would be formed there, discoblastula, as we have referred in some other lecture. Okay, this is how the division or the cleavage would take place in birds. It's meroblastic. In Amphioxus, it is holoblastic, equal. In frog, it is holoblastic, unequal. So finally, when it leads to a blastula, there is a remarkable difference in the blastula stage as well. Let's get to understand that. In the case of Amphioxus, the blastula that's formed, it is coeloblastula. What does that mean? If say this is the stage that we are talking about, there's one layer of cells in the periphery. And there's a huge cavity in the center. The peripheral cells formed by blastomeres, it's called as a blastoderm. This is the blastocele. So a huge cavity surrounded by one single layer of blastomeres. That is what you see in Amphioxus, coeloblastulitis. When I talk about a frog, here also it's going to be coeloblastula, but the difference now is this is the blastocele. The cells in the upper half covering three sides of the blastocele are micromeres, and on the fourth side these are relatively larger cells called megameres. So coeloblastula in frog and coeloblastula in Amphioxus, if you compare, you find this remarkable difference. Here the blastocele is covered by same sized blastomeres. That layer becomes blastoderm. While here the blastocele is covered by micromeres on three sides and megameres on the fourth side. This blastula for frog. The blastula in a bird chick, for example, it is discoblastula. Discoblastula in the sense, say if this is the zygote that has divided, then it will give rise to blastomeres in this region. These blastomeres would then continue and would extend here. So rest of the cell or rest of the sphere is nothing but the yolk, which is not divided. In that case, this would be the blastocele. The cells that are being formed here, in general these are called as periblast. All these cells that are being formed here. These periblast cells would then be distinguished into two layers. This outer layer would be epiblast. The lower layer would be hypoblast. Periblast could be distinguished later on into epiblast and hypoblast. So this is the kind of blastula which is seen in bird. A prominent blastocele, but only related to a disc-like region. So it's a discoblastula wherein there's a prominent blastocele surrounded by two types of cells. That is the blastula seen in a bird, maybe a chick. I would like the videos. Do subscribe to them, share amongst your friends and family, and if you have any queries or suggestions, don't forget to post them. Thank you.